So good afternoon, everybody. Welcome to this year's IDSC Fellows launch event. For some of you who haven't been here before, we have had this program running since 2013, and it's a way to inspire a new generation of leaders in data science to cross with traditional boundaries between disciplines. IDSC wants to join students with faculty, mentors, and IDSC participants, so they work on advanced computing resources in their research. The committee for the IDSC Fellows is comprised by Vance Lemon, Professor Mitsu Ogihara, Professor Kamal Premaradne, Professor Gary Bisham, Professor Mohamed Iskandarani, and Professor Alberto Cairo. So to get started, our first fellow today is Ashley Cook. Ashley is a second-year marine biology and ecology PhD student at Erasmus and at UM studying cetacean acoustics. Is that how you say it? Sorry, English is not my first language. I'm sorry. She worked with Dr. Elizabeth Babak and Dr. Melissa Soldevilla, and is using passive acoustic monitoring to investigate spatiotemporal trends in rice whales' calls and to estimate rice whales' density in their primary habitat in the Northeast Gulf of Mexico. And I'm gonna leave for her to explain all this. So welcome, Ashley.
Thank you. And,
Is the bottom one below me?
you have to share the desk.
You can share the desktop.
Yeah.
Screen it.
Oh,
I can share it.
Oh, there we go. Can everyone see my presentation virtually?
Yes.
Okay, perfect. Hi, everyone. Welcome. Again, my name is Ashley, and I'm going to talk to you about my proposal for using deep learning to look at calls of the Rice's whale, also using passive acoustic data in the Gulf of Mexico. So to start, a little bit of background information about the study species. Rice's whales are in the baleen whale family, and they're the only resident baleen whale found in the Gulf of Mexico. They're actually one of the most endangered animals in the world. There's less than 100 individuals in their population, and in the most recent abundance estimate, there was about 50 individual animals. And they're primarily located in the northeastern Gulf of Mexico in an area known as the DeSoto Canyon, between depths of about 100 and 400 meters. And we know this from vessel-based surveys from the 90s up until present day. So that's what's shown on the right. That study was just from 1992 to 2015. But out of all the effort that's in the U.S. exclusive economic zone, the Gulf of Mexico, and along the eastern seaboard, we really only see them in this area. There's been a few sightings off Texas. In 2017, there was one animal, but primarily located in this biologically important area. And they were listed as endangered in the United States Endangered Species Act in 2019. Originally, they were thought to be a subpopulation of the Brutus whale species, but this year it was determined that they are a distinct species. So they're the new species of whale. And one of the ways that we study whales is using a technique called passive acoustic monitoring. And this is because whales produce a lot of sounds to carry out all their basic life processes, from communication to navigation. And baleen whales, like the Rice's whale, produce a lot of low frequency, high intensity sounds that can propagate over large distances. So that's convenient when we're trying to study them. So we use this technique where a hydrophone or an underwater microphone is deployed in the ocean, and you're able to listen to the surrounding soundscape. And this is useful because a lot of visual-based methods, it's hard to detect animals 24/7. It's very expensive, and you can cover broader ranges using acoustic methods. And what you're looking at on the right, those are spectrograms. So that's a visual representation of sound with time on the x-axis and frequency on the y-axis. And the intensity of the sound is depicted by the color. And rice-pulse produce two broad categories of sounds. The top one is called the downsweep pulse sequence. And these sounds range from about 110 to 48 hertz and about 2 to 25 seconds. And the other sound, which is more common, are long moans. These range from about 150 hertz to 95 hertz, last from 2 to 45 seconds. And they have three pretty distinct regions. It's 150 hertz constant tonal, then there's a slope, and then it tails off. And another benefit of passive acoustic monitoring and studying animals that you can look at trends in calls over time. And when you have enough instruments in a close area, you can use that to estimate density, which is what I'm going to be doing with my research. And we know a lot about the rice as well calls because in the aftermath of the Deepwater Horizon oil spill in 2010, NOAA Southeast Fisheries Science Center, as well as Scripps Institution of Oceanography, deployed 5 hydrophones in the Gulf of Mexico for subsequent monitoring programs on cetaceans in the area. And one was placed right in the middle of the rice as well habitat, which you can see in gray. And from these data, it was analyzed for 8 years. That was coming out in a recent paper, but long moans were detected about 95% of days. The downsweep trains were detected about 31% of days, but this supports that this area is their primary habitat. And another interesting thing that was found was that there's trends in calls. So they tend to be higher rates of calls in the fall versus the spring. And there would also be periods where no calls are detected at all. So in efforts to monitor human activities in their primary habitat and also to look at trends in calls over time, PAM was expanded to 17 sites this year to completely cover that habitat. And as you can imagine, with these long-term data sets, they generate a lot of recordings, and it's not really feasible to go through them manually. So they require robust and reliable automated methods to detect calls. And the problem is the current call detector that we have in place right now has very high false positive rates, and this is due to a lot of the industrial noise in the Gulf of Mexico, like shipping and seismic. So, for example, the long moan detector, this is about 6.5% of calls. This is from a test data set. It has a false detection rate of 26.4%, whereas the downsweep pulse detector has a miss rate of 12.6% and a false detection rate of 69%, which isn't so good. And one of the reasons is it sometimes gets confused with ship noise, and also when the instrument, the hydrophone, is writing over the recordings, it makes this strange disc write noise that gets confused for calls as well. So in efforts to kind of mitigate this problem, and what a lot of bioacousticians have been doing more recently has been turning to deep learning methods for detection and classification of calls in marine mammals. This improves the accuracy and the speed of the detection process and gets rid of a lot of the false positives that's been noted. And it's been studied in a lot of different species. It's been looked at in humpback whales, sperm whales, belugas, North Atlantic right whales. And all the papers that I've read use convolutional neural networks, which are a class of deep neural networks that are very precise in image recognition. However, one of the limitations is that if you're showing the detector an image of a spectrogram like you have in the top right, the answer is only yes or no. It contains a call or it doesn't, and it doesn't necessarily say how many calls there are, which is what I'm interested in knowing. So the goal for my project is to develop a call detector that can distinguish these two call types and be able to say, like, give a timestamp when they occur. And obviously this would benefit ongoing and future passive acoustic monitoring studies. And then in the bottom right, I've done some preliminary work in training detectors. So this was one for a downsweep train detector. And you can see there's a lot less false positives. However, it doesn't accomplish the goal of saying exactly how many calls there are in a recording. So as part of my methods for my project with IDSC, my mentor, Gang, he recommended first using commercial music software. One program is called Melodyne, or also Adobe Audition, for separating whale calls from ambient noise. And essentially these programs can recognize different pitches in recordings. And ideally, I'd like to be able to tease out the Rice's whale calls from the ambient noise to then use in the second step which is time frequency analysis, where you're expanding the one-dimensional calls, the recordings, into two-dimensional images. And these serve as inputs to a call detector. And then the third step would be training the detector using cloud-based deep computer vision systems. He recommended using Google Auto ML. And this is it's useful because it uses transfer learning, where it's already trained on a model, and you're essentially using that model to improve, or you have a preset model and you're using that as a starting point for your problem that is usually similar in nature. And another benefit of this auto ML is that you can create bounding boxes around particular parts of images. So being able to set two calls in one spectrogram, which would solve my issue. So this is my IDSC team. As I mentioned, my mentor is Dr. Gang Ren. He has a background in electrical engineering. He is a research assistant professor at IDSC and also with the Department of Computer Science. And his research mostly centers on artificial intelligence and machine learning, which is obviously very helpful for my work. Then he asked me to mention his partner, who is here right now, Dr. Mitsunori Obihara, who is the director of data mining at IDSC. He has appointments with the College of Engineering and the Frost School of Music. My advisor from UM is Dr. Babcock. She is a fisheries biologist and an expert on stock assessments and ecological modeling, and using innovative data and analysis methods to inform conservation and management of fisheries. And last but not least, my NOAA advisor is Dr. Melissa Soldevilla. She has a background in biological oceanography. She's the PAM program lead at NOAA Southeast Fisheries Science Center. And our research in our lab uses passive acoustic monitoring to study marine mammals and help improve management and conservation. And that's me, obviously, through this program. And from my mentor, I hope to improve my understanding of deep learning, because right now it's still very mystical to me. I'd like to understand a lot more of the math behind it. I think that would be helpful, learning about the high performance computing resources at IDSC, and also learning how to use cloud-based deep computer vision systems. My courses, and that's it. Does anyone have any questions?
Thank you, Ashley. So now we have the room open for 5 minutes for questions from the audience.
Yeah, I have some.
Okay.
Sorry, I don't know whether I'm allowed to ask a question, but so you said counting the number of calls in one recording. So do those say multiple calls? Do they overlap, or?
Sometimes they do. So like in the image, there might be one call and then one very close next to it. So that's kind of the problem that I've been running into, is how to deal with that.
I see. And you mentioned the ship noise.
Yeah.
Are there any noises?
Like other noise sources in the Gulf. Yeah, so the ones that we mainly get are shipping noise, which is low frequency, and then there's a lot of seismic noise from seismic exploration in the Gulf, and then other commercial shipping, and then the disc write noise. So are the big ones.
Okay. And I'm guessing that the human, a trained human being able to tell how many.
So when you would do that, you would look through, while you're listening to the sounds, look through the spectrum, go across the screen, and be like, that's a call, that's a call, that's seismic noise. So it's a visual problem.
I see.
Yes.
Okay.
Yeah, there's something
And
about the
sorry, I have one more question.
Go ahead.
How about the individual variability? So you have two, right? A whale, the same species.
Right.
How similar are they both?
They're pretty much all the same. So you'll see it has, sometimes they do mostly differ in length, and the signal strength that we get depending on how far the animal is from the actual detector. So that's really how they vary. But from my preliminary analysis, they're all very consistently like within that frequency band, and just the times are pretty variable.
Okay, thank you.
Yeah, there's one small point about the noise in the recording. So we find because there's silent regions. So right now, Adobe Audition offers option. You can first get in the noise profile from the acquired segments with the machine noise, with both noise, but without a whale sound. And then you learn the characteristic of the noise, and then you apply the noise profile to the complete recording. So we find that function can remove those noise quite effectively. So we are thinking maybe next time we are using similar functions from Adobe Audition or Dataset.
Okay.
Thanks.
So we also have a question from Professor Ruiz Pestaña.
Yeah, hi Ashley, great presentation. I was curious for the classification problem, you need, I imagine, a label data set. So is there anything like that in the field? Has somebody been looking at recordings of this frequency time and identifying whale calls that you can use to train the classifier? Or is it something that you want to be working on to create like a large data set that people can use as a benchmark for this problem?
Well, so I guess to answer your first question, for training the detector, I forgot to mention this. So I do already have someone in our lab when they were creating the first detector has a CSV file of all the start and end times. So we have an annotated list of when all the call times occur. So I've been mostly working with that, and kind of to answer your second question of like the purpose for this, obviously it would be helpful in all kind of passive acoustic studies focused on this one particular species because you're training on that one call type. But it's been done before in other species of whales, just mostly looking at presence, whether or not they're there or not.
Thank you.
Mm-hmm.
Thank you, Ashley. So we're gonna go to our second new IDSC fellow. His name is Julian Dallmeier. Meyer. So Julian is a third-year neuroscience PhD candidate under the supervision of Dr. William Scott. He's currently researching clearance mechanisms of Alzheimer's disease associated pathology at the Brain Endowment Bank. And I'm gonna leave him to explain his project.
Okay. Let's see. Yeah, the bottom right was hers. Oh, this is. Oh no, this is not. Yours is in the top right. Okay, okay. I've used the same template before, so I was like, oh, that looks familiar, but I forgot I switched it. You're good. Click the button in the bottom. Oh. Max. Okay, hello everyone. My name is Julian Dallmeier. So my IDSC fellow actually decided to join us in person today. And like I mentioned, my dissertation mentor is Dr. William Scott, and I'm doing my research at the Brain Endowment Bank, which is over in the Life Sciences building on the medical campus. And the project that we're going to be working on is spatial clustering analysis of corpora amyloidosa and tau in the hippocampus. But before I get started, I just wanted to say I think this program is a great opportunity for young scientists. I remember joining the university about 2.5 years ago, and I saw this program, and I knew I wanted to be a part of it. So I'm really honored that I was chosen, and so thank you everyone for believing in me. So that being said, I'll give a quick background on Alzheimer's disease because that's essential to understand what we want to do here. So essentially what we want to do is explore the spatial accumulation of Alzheimer's disease pathology. So Alzheimer's disease, as most of you are probably aware, is the most prevalent and destructive neurodegenerative disorder. And clinically, it's characterized by memory deficits, but it can actually present itself in other ways and cause other issues such as emotional imbalances or issues with executive thinking. And a lot of that is strongly correlated actually with the location of certain pathological markers. So depending on where some of the pathological markers are found, depends a lot on what kind of symptomology that patient will have. So generally, when we're looking at Alzheimer's disease brains, we see a lot of buildup of hyperphosphorylated tau and amyloid plaques. In this project specifically, we're going to be focusing on tau because it's more correlated with the cognitive deficits seen in AD. So we believed it to be slightly more important. And then actually in my dissertation project, I'm looking at a potentially novel clearance mechanism called corpora amylacia, which are essentially cellular waste bins, and they clean up this pathological debris and export it from the brain. So the kind of data we have at the brain bank is, so we have a clinical background on all the brains that we received there. So there's demographic data, sex, smoking history, things of that nature. But on top of that, we actually also have genetic data sets. So we have whole genome sequencing for a lot of the brains as well. But more importantly for this project, we have immunohistochemical stainings and tissue samples from human brains. So what we do with that is we harvest the brain from a donor, and then we section it and cut it into very thin slices where we apply different reagents or antibodies to try to paint a picture of what's going on and stain different cell types and different organelles. So on the left here, this is an H&E image, hematoxylin and eosin, of a human hippocampus. You can see this dark cell line in the middle. That's actually the dentate gyrus area. And this is a very interesting part of the brain because there's a lot of projections in and out of this brain. So it's a perfect example of a complex neural network where this part of the brain is connected to many different areas of the brain. So it receives a lot of projections and there's a lot of inputs and outputs that happen there. So when we zoom in, we can see different details. So these images are massive files, hundreds of megabytes, and you can just zoom in and start to see different cell types. So on the right on the top, this is an example of corpora amylacia, which I mentioned earlier. They're kind of these dark moon-like structures. And then on the bottom is an example of tau. So on the bottom left, you can see an Alzheimer's disease brain. And there's all that brown stuff is tau. So that's hyperphosphorylated tau. And then on the right, the figure I, that's a normal control. So you don't really see any brown staining there. So it's a dramatic difference between AD, severe AD, and a healthy control. So the question that we have with combining our resources and of course the resources of IDSC is, does phospho-tau or p-tau, do these aggregates and CA cluster in specific areas of the hippocampus? And is this clustering associated with increased genetic risk? So essentially right now, how neuropathologists characterize AD is a semi-quantitative measurement of tau. So it's essentially presence, location, and then a low, medium, high semi-quantitative classification. But we believe that there's a lot more than meets the eye, and that by looking at where they're clustering and different patterns inside the brain, we can uncover more information. So the approach we have for this project is, well, first we have to get the data. We're in the process of doing that right now, but we're going to start with a preliminary cohort of 15 controls and 15 AD patients. And we'll take the hippocampal human postmortem brain tissue, section, stain, and digitize it for computational analysis. And then we want to extract some features from these pathological images and create new features, so new data from the existing slides. And still discussing with Dr. Ogihara how we want to do that, but some potential options are some dimensionality reduction techniques. And then we're going to move on to clustering, where we will perform clustering algorithms and to try to analyze the distribution of these, both tau and corpora amylacia. But in order to do that, we will potentially use k-means clustering or nearest neighbor analysis, or potentially also mean shift. But again, we're still working on the fine-tuning the details of that. So here's the team. So of course, me, the IDSC fellow, my mentor, Dr. William Scott, who is a professor at the Department of Genetics, but he's also a part of the Department of Neurology and Neuroscience. And he's got a lot of experience with epidemiological studies, genomic analysis, and as well, he's also the director of the Brain Bank. And then Dr. Ogihara was kind enough to join us in person. He's the director of IDSC Development and Education and professor for computer science. So we're very happy to have him as a part of our team, and I'm looking forward to working with him. So the project goals that I have for this, first I want to learn. Learning is the number one thing. It's not often you get the opportunity to work with such renowned scientists. So I'm very looking forward to work with Dr. Ogihara and learn more about methodology and data mining and feature extraction. And also, understand the algorithms, not just how to use them, but more what's going on under the hood and some of the mathematical computation behind that. And then discover. So it would be great to, unveil some novel distribution patterns and associate those with different genetic risk factors. And then finally, it would be great if, if this led to a publication or an interdisciplinary paper, just to really show the fruits of this IDSC collaboration. So yeah, just a quick timeline. Right now we're working on getting the data and fine-tuning exactly what methodology we want to use for this project. And then we'll start going from there. So that's it. Any questions?
Thank you, Julian. Any questions?
No, I don't.
Okay.
Do you think you're going to use the images in part of the learning process?
Yeah, when I was writing, so that's a good question. When I was writing this proposal, I was thinking, you mentioned convolutional neural networks, and I was thinking maybe there's, a place for that, but I'm not sure.
Yeah.
Potentially. It might be easier to, I don't know, we'll see.
Just all that like extensive data just on the record.
Yeah, it's there. The images are massive, so I don't know how that would work with the convolutional neural networks, because the resolution is very high. So
Julian, there are a lot of papers looking at neural networks as a way of classifying pathologic data, as opposed to trying to quantify the pathology that you're interested in and looking at the spatial arrangement. So we could take a look at that and see if there are any aspects of those algorithms that could be applicable to what you're doing here.
yeah, and exactly, that's correct. The CNNs are great for classification, but in this case, we know it's an AD brain. We don't need to classify it. So, but yeah, there might be something there, but we'll see.
I have a question.
Yeah.
So is the goal then to kind of look at, like the quantitative measure of Alzheimer's and see if that correlates at all with any genetic risk factor? So I guess like if you have both the brain bank as well as do you have the data set, both the brain slice and some genetic markers that you have for them that you could do
Yeah.
that?
So the cohort we selected actually is paired with the genetic data. So we did that on purpose so that we could kind of go back and say, so the idea is to kind of look for distribution patterns and then correlate or associate that with genetic variants. So maybe you have a genetic variant that, increases your sequestering of the pathology, and that might be a beneficial thing. And maybe we can see that very clearly in people that have that genetic variant.
Another side question.
Yeah.
I have no idea if you have this data, but I'm just thinking really cool. Do you also have like the medical history for these patients? So for example, whether or not they were taking different types of drugs,
Yeah.
to see if that had an effect.
Yeah, now we do have that, but I've tried to look at that in the past, and the issue with that is that it's very hit or to have a large amount of people taking the same drug for the same amount of time, you don't have the end
Very
number
sparse.
right there, exactly. And then the other issue with that is that a lot of this data can be got from next of kin, from their parents or their kids. And the reliability of that data, it's not as clear cut as like a genetic data. So we try to avoid using the clinical data except for sex, age, and things of that nature. But we do have that if we ever find it, we want to look a little bit deeper.
Okay. So I think given the number of samples, which is kind of small, The bringing in or adding more confounding, possibly confounding factors could sort of make the analysis blurry. So probably we should rather go with a very small set of factors. I mean, people.
And get more information
Right,
on each subject,
exactly.
exactly, instead of, yeah, and we have the means to increase the sample size if needed, so that's
Right.
it's always,
Right.
always an option.
Mhm.
I saw there was
Thank
another
you.
question there, no, okay.
Yeah, I saw Professor Lewis had his hand, but he lowered
Yeah, I
it.
just didn't want to take any more time, but I was curious to see, now that you asked me, I'll ask. I was curious to see if there is any characteristic size about the aggregates besides the spatial distribution. Is something you expect to increase just with the I don't know, age or other factors, or is something that doesn't matter as much?
Great question, yeah.
Related to the, I guess, multi-resolution, right? Where are you looking aggregates? At what size? Why which ones are actually relevant for the pathology? Thank you.
So yeah, that's a great question. We proposed that not in this particular project, but we did want to look at the different size because, you know, that since they do sequester trash, you could assume that the bigger they are, the more trash they have inside of them, right? So that's something I think that we could probably extract that feature too and add it to our analysis. Yeah. But yeah.
Thank you.
Yeah. Okay.
Thank you. Okay, so now our next fellow is Kevin Davis. Kevin is a second-year graduate student in the Medical Sciences Training Program in the Department of Biomedical Engineering. His research centers around neuromodulation and rehabilitation for movement disorders such as spinal cord injury and paralysis. Go ahead, Kevin.
Okay, thanks. Yeah, so I'm really excited for this opportunity to be a part of the IDSC, kind of similar to what Julian said. I've seen this before, and actually this is learning more about machine learning and data science was actually a primary reason that I was looking at biomedical engineering and data science is something that I really wanted to learn more about. So I'm excited about this project, and our goal is to use the computational efforts that were given to expand the functional control of a fully implanted brain-computer interface that we have recently developed. And we actually deployed this to the home of a patient with spinal cord injury, and so I'll get into that. So I want to kind of motivate this by talking about what brain-computer interfaces are, how they can be helpful, what we've done so far with them, and then how this project will help boost our current work. So basically, brain-computer interfaces, in the most simple sense, can be thought of as anything that takes data from the brain and then translates it into something useful for a user. And so in this capacity, it has quite a potential benefit for individuals who have some sort of neurological loss from injury or disease. If we think about a healthy human patient, with their desire to move, movement intention begins within the motor cortex of the brain. It travels through the spinal cord out to the muscles and allows us to control our limbs. But after injury or disease such as stroke or spinal cord injury, the connection between the brain and the muscles becomes lost. And so brain-computer interfaces sort of get around that disruption as we take the data from the brain and decode what the subject was intending to do, and then can reanimate that using like a neuroprosthetic or an exoskeleton. But this is just one component of what brain-computer interfaces can do. They've also been demonstrated to do things like speech synthesis for individuals with amyotrophic lateral sclerosis, Lou Gehrig's disease, as well as sensory prostheses. So things like visual or audio, or in fact, cochlear implants are a good example of an auditory brain-computer interface. So ideally, these type of systems for paralysis would be a clinically viable tool. However, the current methods that we use for sampling data from the brain limit this ability to take it from the research environment into community and home setting. And this is largely driven by the methods that we have from collecting the data. So on one end, we have invasive methods. This is like an intracortical microelectrode array. So this is like a little chip that has a bunch of electrode spikes or shanks. that are driven surgically into the cortex of the brain. And this is nice because it puts those electrodes right up against the neurons and has excellent signal quality. You do a lot of analysis with that data. But this comes at the cost of having the subject directly tied in to the recording hardware. And so they're limited to the research setting. Non-invasive systems such as electroencephalography, which are electrodes placed on the surface of the scalp, are great because they don't require surgery, and so there's many more people who can be involved with using EEG. But in this case, the signal has to traverse the skull, the scalp to get there, and so there's decreased signal quality. And on top of that, caregivers who need to be setting up these systems in the environment, in the home environment, find them very difficult to set up. It's pretty lengthy. I think the most recent paper demonstrated it was about 20 to 30 minutes to set these things up. And then you also, how much function are you going to be able to get out of that? So this was our first approach at establishing just a baseline brain-computer interface for the home. We decided to go with electrocorticography. So this is still a surgical procedure. In this case, the electrodes are placed on the surface of the brain. So what we have over on the right is the configuration of our electrodes. We have placed these electrodes on top of the motor cortex that corresponds to where the patient's desire to move their dominant right hand is located. And then these signals are collected in 4 different channels. That data is then sent to a mini computer that we've placed on the back of the patient's wheelchair. And then it decodes that information. And right now it's a simple decoding algorithm that just looks at opening and closing the hand. And then it sends that command through Bluetooth to a little mechanical glove that they're wearing. So it allows them to open and close that hand now. And with this system, we've demonstrated with their caregiver that they're able to set this thing up in about 5 to 6 minutes and be able to use this. And so as part of my main dissertation work, we're looking at, okay, well, with a system that is as simple as this, how much are we actually able to derive with this in terms of improving their quality of life and re-enabling them to engage in activities of daily living? And so that's one of the questions that we're looking at. But for this project, we want to see how we can improve our decoding mechanisms and functions of this. And so to show you what we're doing so far is this is data from the 4 channels that we get. So it's primarily 2 time series channels and then 2 average hour channels. And what we've done is we've given the patient an app on their iPhone where they can sit and they're presented with a prompt, such as currently just move and rest. And so as the data is coming in, we label it with this prompt that they're seeing. And then as it's coming in, we window this data and then we can create a feature vector or a list of features from there, the spectral features. And to show you how this actually differentiates, if you look at the bottom plot between channel 1 and 3, when we look at the spectral features here, we can really separate out between the rest and move. And then we take those feature vectors, throw it through a simple signal decoder to look at rest and move. And so that's currently what we're doing. This is actually how we collect the data. So I should also note that this, all of the data that we have right now is all collected from within the patient's home. And this allows us to collect a lot of data. The patient, basically whenever the patient wants. So in fact, most of this data was collected in the evening for a long amount of time. So this shows how that the prompt looks on the patient's phone. And so this shows that what we've achieved so far is a very simple home-based binary motor imagery classifier, which allows him to open and close his hand. But recent surveys that have been published in the literature have shown that potential users of BCI would really like to have lots of function in any system that they have, whether it's for rehabilitation or assisted. And so our system really lacks robust control functions. And so it's really just this binary signal. And much better to either have more classification or even regression on different things that we're trying to do. And so this is where our project comes into play. And so this is our team. So myself, I've done most of the software development for our system. I have a bachelor's in neuroscience. My professor, my primary mentor, Abhishek Prasad, has a lot of work with motor control, as well as neural interfaces in both humans and animals. And then our IDSC collaborator, Odelia Schwartz, who's already, we've met with, and she's provided great insight, different things that we might be able to do with this data for looking at deep learning and classical machine learning algorithms. And so our project goals, we have 2 of them. The primary one is just to add additional motor imagery states using this ECOG signal. The secondary thing that we want to look at is predicting real-time position and velocity kinematics of a computer cursor using this ECOG. And so this is a brief tentative timeline where we're going to look at this first goal of motor imagery states where we start looking at protocol design, where we start collecting data, and then training that. And then once that gets going, begin to start playing around with looking at 2D kinematics. And so how we do this is looking at additional motor states. We currently have open and closing grasp of the hand. We want to add on top of that things like wrist flexion, extension, ulnar radial deviation, as well as elbow flexion and extension. And as part of this, look into what are some of the signal processing things that we're doing as potential hyperparameters for our decoder accuracies. And then below, this figure just shows what this might look like. We recently wanted to play around with additional motor states, and so we had the patient actually thinking about left hand versus right hand movement. And so this kind of shows you what the signal will look like as it's labeled, rather than just a binary black and white, but different classes. And then for the secondary goal for position velocity kinematics, in this case, rather than just simply showing them a prompt, we'll have them have a cursor that will then move to various points along the screen and have them think about moving that cursor themselves and then attempt to regress that neural signal and those features with the actual 2D position as well as the intended velocity. And here's where we really want to look at a lot of the different classical machine learning techniques, as well as deep learning algorithms that Dr. Schwartz has suggested we look at. And if we have this regression, this will not only provide more function, but also fine-tune control. And I should mention that this stuff isn't novel in terms of the decoding, but is novel in the fact that we're using basically 4 channels or 2 time series channels and wanting to squeeze out as much function as we can from those using the algorithms, which would essentially show that we have technology currently available to put this in the home of these patients to demonstrate a proof of concept that this technology can be or closer to potential assistive device deployment. And so the expected outcomes of this is primary to improve that BCI system that we have from use. And then the second one is so that I really want to gain a better appreciation for how we can extract insights from large data sets, how we can develop good, robust decoder architectures. These things are, in my mind right now, are kind of a black box that I'd really like to learn more about. But then on top of that, as I gain that understanding as a future clinician scientist, be able to understand how I can collaborate with other data scientists for future clinical endeavors on how we can use that to really benefit patients in the clinic. Thank you.
Thank you, Kevin. Any questions from our audience? Icebreakers. Professor Ruiz. Just kidding.
I think my question, so I have a question about the purity of the data. Meaning, how about the individual sort of variability? We collect the data from multiple people. Is the data sort of dependent on the exact location that you put the device in?
Okay.
Meaning if there's slight difference,
Yeah.
right? The location would there be a significant difference in the signal, the 4-channel data you're collecting?
Yeah.
Also, my second question is sort of purity. You have this interesting, I think, very natural, a method for using mobile phone to instruct the subject to think about right, the movement. So my second question is, how good do you think this method is?
No, this is great. So the first question with, in terms of subject generalization, right? So the method that we're using right now for the open and close stuff, that has been known since the 1940s by Jasper and William Penfield. They were neurosurgeons in Canada that were looking at this. And so, and it's called event-related desynchronization. So the idea is whenever you start to move, no matter what it is, there's going to be a general decrease, generally, in the beta band area, which is about 13 to 30 hertz. And so that is generalizable. Now, whether or not, more fine-tuned things are generalizable, that's still to be looked at. The other thing to note in terms of the quality of the data that's important is the frequency spectrum is often divided up into alpha, gamma, delta, theta waves, right? And there have been papers that have shown that the high gamma and low gamma area is really helpful for decoding movement kinematics. And that for us right now, so we're sampling at about 200 hertz. So right, we're barely breaking the cusp of that. So we really want to see if we can take advantage of the data that we have to drive this forward. So even though we have some of that cut off simply because of the microwave frequency, although we have the top portion of that gamma band, is utilize that top of everything. Now, whether or not that will translate is a question to be answered later. The second question of, is our method actually, right? We have the patient looking at a phone, and the phone is actually Bluetooth, right? So you could ask the question of, how is the temporal quality of this thing actually working? Because you have to think about the reaction time of the patient. When does the computer actually start labeling the data, and how fast does that prompt get from the computer when it's labeling to the phone? And that's actually something that we're hoping to publish soon, that we've kind of benchmarked and profiled this software to demonstrate what that might be. One of the original things that we started looking at a couple years ago, or sorry, a year ago, when we started putting this in the home was, does the current decoder accuracy, which is binary classification, get better if we delay it a little bit, assuming that there is, we need to account for both patient reaction as well as, the label to get the phone. We found out that there's actually not, actually decreased the accuracy. So we think this is actually pretty good. And the time to actually get the label to the phone is in milliseconds, whereas these reactions, very small milliseconds. So, I mean, we think that this is actually pretty robust method to do in the home to get the data. And so we're hopeful that this is actually good.
I see. Okay, so now I have then another question. Is there a reset sort of command to it?
So what do you mean?
So before moving hand, right?
Yeah.
So can you tell the subject to do something, think about totally something different, right? So like say drinking water.
Okay.
So watching movie. And will that help into annihilating everything that has occurred before? So you can have a fresh start.
I see. So are you talking about, just to clarify, are you talking about resetting the decoder, or are you talking about like how he's going to think about these movements in the environment of distractions?
Right. So I think it's both, right? You have some residual, right, sort of brain signal that is, say, being recorded.
Right.
So by the time your signal, like, the instruction comes in, there may be some residual movement sitting in, right? So that's
possibly.
Right.
Yeah. And
so I was wondering if there is something reset.
Right.
You can.
Oh, in case the data needs to be
right.
rolled.
Exactly.
Yeah. So the phone that they actually have gives them full control. So we use the phone that they have as both a device for them to control the system, because we did it in a way where their caregiver and the patient are pretty much running it themselves. Like we can instruct them. We could devise a way where we're controlling it remotely, but it actually is easier for them to know that they're in control. And so they can actually, in the middle of the session, if the subject said, oh, you know what, I got distracted, you can actually click back and just
okay. Sorry. Thank you.
Yeah.
Question.
Yeah. And actually on top of that, what we're able to do, because all of our code is based off of the GitHub repository, and so every time he turns it on, it just pulls everything down. So if we want to add new things that he wants to do, we just push it to the main branch and he starts it up, and now it's there on his phone because it's dynamically displayed.
Thank you.
Yeah.
Thank you, Kevin. So now we're going to our last fellow, Mingyue Wu. It's currently a first-year PhD student in the Civil, Architectural, and Environmental Engineering Department, following Dr. Luis Ruiz Pestaña. Her doctoral research focused on predicting the aging behavior of colloidal glasses based on molecular dynamics, simulations, and machine learning techniques. Welcome.
Thank you, everyone. Hello, everyone. My name is Mingyue. I'm from Civil and Architecture Engineering Department. So for most of my project, I use physical-based molecular dynamics simulations and machine learning to predict the mechanical behavior of the concrete clay. So I think we are very familiar with the concrete, which is the gray material we use to build our buildings. But the concrete gradually attract people's attention because as it is described by the news, the concrete is defined as the most destructive material on the earth. This is because during the process of producing those concrete, we also release a huge amount of carbon dioxide into the atmosphere, and which will cause the global warming and the greenhouse effect. So we need to come up with some strategies to eliminate the environmental impact of the concrete. So the first strategy is to develop the green alternative materials like the bioconcrete, or we try to recycle or reuse the waste concrete materials. And the third strategy is correlated to my project is to enhance the concrete durability. So imagine if we could increase the lifetime of our concrete building from 50 years to 500 years, then we do not need to produce too much concrete, and then we could release the carbon dioxide emissions. So creep is one of the main factors limiting the lifetime of our buildings. And creep is defined as a long-term deformation driven by the sustained load. So at the animation show here, if we apply load to a system and last for some time, and by the end we release the force, we could see there is a permanent deformation in the material compared to its original shape. And those two pictures in the right-hand side, you could see that is the real creep phenomenon happening in our daily life. So if we want to better deep understand the creep behavior, we need to transfer our attention from those big concrete bridges to the nanometers of the cement matrix. And in the cement matrix, there are a lot of nanoparticles, they move. And if we apply the load to and some nanoparticles, they will move in a very long distance compared to the other nanoparticles. And this is what actually causes the creep. So actually how much a given nanoparticles will move and where it's going to move actually depends on the structure of its local environment. And here I need to say that this is actually in the nanostructures, the cement is actually the glass. So in the next following step, we're talking about the glass systems. Okay, so here comes to the most challenging part of this project. It is the problem that we do not understand the structural property relationships in the glass. So in the glass, the structures of the liquid and the solid are very similar. However, the properties such as the viscosity, they could increase by 14 orders of magnitude. So if we turn back to our previous topic, we need to know the structure and the dynamic properties in the glass. And it is proved that the simple structural matrices in the glass, they are only weakly correlated to the dynamics or the mobility of the nanoparticles in the glass and the load. So in the project, we want to design the resistant cement nanostructures. And the problem here is that we do not, the simple structure matrices do not really work, and we do not really know what we are looking for. But there must be something encoded in the glass structures. And machine learning here, they provide a very promising method because it is really good at recognizing the complex patterns. So we want to use the machine learning techniques to gain insight into the great behavior of those glasses. So here shows my research plan. First, we want to generate enough data from the molecular dynamics simulations. And then we use those simulation outputs to calculate the complex structure matrices. And we put the structure matrices as the features and the output of the simulation as our target to train the machine learning models. And we want to predict the particle movement in the glass as a function of time and with different applied load. So here comes to my goals of this project. First, we want to identify the structure matrices that correlate with the great behavior, and by the end, we want to predict the glass nanostructures that are correlated, that are very resistant. So here shows my team. My academic mentor, Dr. Luis Pastana, who is an expert in the molecular modeling and simulation. And my IDSC mentor, Dr. Melvin Ayala, who is an expert in data mining, AI, and machine learning. And for me, I have 3-year programming experience and 1-year molecular modeling experience. And besides that, I'm also of course, I'm modeling expert. So maybe you heard my news in the UN news, and that showed my previous experience at Westmas. And what I want to learn from this project is to learn how to manage the big data, because we are going to generate millions of data points from the innovation. And second, I want to learn more about machine learning techniques such as neural networks and how to use the machine learning to treat such a huge big data is also a challenge. And thank you for listening.
Thank you. Thank you very much. Any questions? We have at least 3 to 4 minutes for questions.
So in your simulation, so how many molecules are the scale of the simulation model?
3,000 items for each simulation.
Ah, okay. Quite big.
Yeah, well, so for each simulation, so actually in our, we are going to have 500 simulations. So that's why it's big.
I see. So I'm guessing that in your simulation, each molecule sort of interacts with the neighbor.
Yeah.
So on average, how many neighbors that each molecule interact?
Yeah, so around 13 to 14.
Ah, okay. That's quite big.
Yeah, it's one thing. So that's why
Great.
we use a simple structure to predict their movement. Their movement depends on their actual space between its neighbors. If the neighbor they are close, they can't move to that way. They need to move to another with wider space compared to its neighbors.
And how do you visualize your model? Right? You're going to use a molecular simulation, right? To generate, say, this is T0, and you have this delta T, etc. There's now, is there a sort of ground truth data that you can use to sort of check how accurate this sort of model is? Or say, are you taking some, say, a simulation model that someone has already developed for this purpose?
Yeah. So actually, we use a special graph model.
I see.
So it's a lot of people use researchers they use that model, so that's why we choose a very special model.
What is the output of the model? So you're getting
So that is so
the average interactions.
different time, how each particle in different positions. So it's X, Y, Z positions.
You're getting X, Y, Z of all the molecules.
Yeah, of all the
Okay.
molecules.
A lot of data.
Yeah.
Cool.
Thank you, Mingyue. So with this, we finalize our information of our 4 fellows. And I would like to thank you all for joining us and wish a great project for each of them. And we'll see you again in April with the results.